Hi there, Greg. It's so great to have you here live with us today. Fantastic talk. It's so great to hear about the different roles you've taken up in your career, and thank you so much for sharing that.
You're welcome. I'm delighted to be here, and I hope it's of some use to people.
So we have some questions coming in from our leaders of tomorrow who are super excited to quiz you on antibodies. And so the first question is, what role do you see antibodies playing in the treatment of COVID-19?
Well, I guess there's two types of roles that they could play. The first is in dealing with the inflammatory reaction that takes place and is the main killer of old people in particular. The second is to try to block the virus. If we look at the inflammatory, the use of antibody anti-inflammatories, there are a number of inflammatory targets that could be addressed with antibodies and are being addressed with antibodies, but so far they haven't given unequivocal answers. One of the issues with this disease is if you've got a patient who is infected and taken into hospital, then if you give an antibody that's essentially an anti-inflammatory, it's the same with giving any anti-inflammatory drug, you're in danger of shutting down the body's own response to the virus, which would kill the virus. On the other hand, you want to block the response, which is actually over the top. So it's a bit of a fine balance. So what you need to do, the issue with antibodies, or actually any other anti-inflammatory drug in treatment of COVID, is the point at which you're going to give it. And that's very difficult to manage. It would have been easier, in fact, maybe it'll be easier now, but there's been a period for which, for example, it takes a while to get antibodies to be approved or to be agreed that a clinical trial can be run even with an existing approved antibody. And so I'm aware of at least 2 or 3 trials where people would like to get them into patients, have had to stand by and wait because actually there aren't the patient suppliers dried up. I mean, during the summer. Of course, we may now have enough patients coming back with the return of the disease. So I think we will expect to see data coming in from antibody anti-inflammatories. There are a number of targets that are being looked at, TNF, there's also antibodies against C5, which is a complement reaction, which is involved in this major inflammatory burst. So the other use of antibodies would be in blocking the virus binding to its receptor on cells. So these would be antibodies against the receptor binding domain. And a number of such antibodies are in development and may well be in early stage clinical trial. I think some must be in early stage, just toxicity trials at the moment. There are antibodies being made by, for example, AstraZeneca. The strategy there has been to take, is because people think the virus will mutate, it could be advantageous to use 2 antibodies. So therefore you're covering 2 possible epitopes. And the aim would just be to stop that virus binding to the receptor. And my guess is that they will have their role to play. I think in particular, in treatment of older people early on. And the reason I say it is because I think there's a very good chance the vaccines, older people don't always react so well to vaccines. And therefore it may be better to put in an antibody against the virus once you know they're infected. To try to dampen down the viral replication, try to dampen down actually its entry into cells. So if you can do that, that would certainly be good. It might even be possible to use it like a prophylactic. So perhaps for in care homes, for example, to give protection for a couple of months by injecting an antibody as the virus flares up. And that would be great. But the problem is the scale of creating the amount of drug that you need. that, potentially there's 7 billion people you might need to give the thing to. And I mean, even in the UK, if you had millions of doses, it still wouldn't be enough for dealing with the people who are vulnerable. When you take into account old people, people with diabetes, etc. So it really is, I think antibodies will do some good in those two different roles. There's absolutely a case for them. There's probably a case for two together being used to try to avoid the creation of or the evolution of viruses, which would escape that selection pressure. But I think we just need to have lots of different drugs from lots of different sources, and we need to isolate as much as possible. We need to use vaccines, and we will probably need to use antibodies and small molecule drugs. I mean, we've got to try a wide range of approaches and get these things into patients fast.
Absolutely. Thank you for that, Greg. So next question is about your career journey. Having held a number of different positions during your career, what are the most important lessons you've learned, and how can leaders of tomorrow apply these to their careers?
I'm sort of reluctant to give. I've tried to explain my own career where I adopted an approach which was relatively unplanned. So I never sat down and thought, what do I really want to do? I didn't worry about it. I just got on. Perhaps the most important thing was I tried to focus both scientific work and the other work on things that I thought was important. And of course, it depends on how you define important. My own supervisor many years earlier had said, I'd commented that an experiment would be very interesting. And he said, bugger interesting, is it important? And I thought it's a very profound comment, because there's so many things in life are interesting. There's so many things one can be curious about and wondering how things work. You could spend an entire lifetime doing that. And that would, in a way, be the life of a scientific dilettante, addressing all those little curious little things. And sometimes you'll discover something fantastic by doing that. But actually, I think it's probably better to focus on those things that you think are important. So I've always tried to focus on a big problem, a hard problem, to try to be creative about it, to sort of try and think outside the box. But you've got to be able to combine the thinking out of the box with the ability to deliver. So actually, there are some people who actually have tremendous ideas, but they don't have the technical capability to work the technology or the things that would be required, and they get bored because it doesn't work first time. And another good bit of advice I had, certainly in science, was, well, if it should work and it doesn't work, then you've probably just got to fiddle with it a bit. And that was advice from Fred Sanger. And I think that was very good advice, because quite often, things don't work for some silly technical reasons. And if it really should work, then it's usually by playing around with conditions, or you've got to be self-critical. You don't want to just bang your head against a brick wall, but you've got to keep thinking about, have I got all the controls in there? Try to dissect down where the problem is, and just keep picking away at the problem. So I would say, by focusing on important problems, by being prepared to put the grunt work into learning a technology, and I made sure, for example, my entire career, I was quite lucky, was based on protein chemistry, which in fact became clear very quickly that I was going to be redundant if I just focused in the early 70s on protein chemistry, because recombinant DNA technology was just coming in, and I could see that was going to replace it. But that interest in protein chemistry made me, it was very useful later when I decided that I wanted to use recombinant DNA technology to change proteins. But I had a deep knowledge of protein chemistry and a deep knowledge of recombinant DNA technologies. I learned it by sequencing genes, sequencing proteins. So I probably spent the first 5, 6, 7 years of my career doing very, very hard work. It wasn't hypothesis driven particularly. It was basically, I wanted to get under my belt the ability to be able to control, really understand and know all the little ins and outs of technology. And so I would say that's something, if you're going to be managing a group of people, you want to know more than anything more. You want to know much more than they do. You want to be able to deal with their problems. In recombinant DNA, I was lucky because recombinant DNA continued to be an advancing technology for many years. And so a lot of that early work I did, even when I was no longer doing practical work, people would show me their gels and such like, and I could look at them and I could immediately see what the problems were, or I could identify and make useful suggestions. And that's because I'd had such a, probably 5, 6, 7 years of being right at the coalface under immense pressure, trying to get things to work. And I think that there is, well, in my case, there wasn't a fast route. I wasn't so intellectually superior that I could just do things without any effort. It was incredible hard work and incredible focus on making sure you understand all the nuts and bolts. And I'd be saying the leaders tomorrow, make sure at least that you understand 1 or 2 major areas in great detail, completely become a master of those areas. You can then use that as a firm foothold to go out and look at some other areas. So there's other areas where I, like immunology, a lot of people think I'm an immunologist. I'm not at all. I'm essentially a recombinant DNA guy. I feel most comfortable in dealing with recombinant DNA technology and the potential of that. But on the other hand, I had to get to grips with immunology because that's where the important problems were. It was most relevant for the work I was doing. So I'm trying to think if there's anything else I can advise, but I think it's that mastery of, well, the approach I followed was the getting mastery of the technology and really understanding what I'm doing. So, I mean, if you're an accountant, you'd want to do the same thing. Really make sure that you've got a good grasp of what it is you're doing, really understand balance sheets. Then once you've got that as your core area, you can advance from that.
Thanks, Greg. That's some great advice for our leaders of tomorrow. So the next question is about novel drug modalities. So therapeutic antibodies were revolutionary when they were first discovered. We're seeing an increase in novel drug modalities, for example, cell therapy. What do you think is the next disruptive technology?
Well, I'd like to say it's going to be bicycles, which is the next thing we're doing. So in other words, the reason I'm so excited about them is it does give us these cyclic peptides are somewhere in between small molecules and antibodies and combine some of the advantages of small molecules with those of antibodies. And I think there's a lot can be developed there, and they can be evolved very, very easily by using all the standard methods that we know. But putting that aside, I would say that cells will certainly become one of the important modalities, but don't underestimate the difficulties there. In the mid-90s, we thought that DNA vaccination was going to come in very fast because people were looking at putting viruses into cells and actually just putting DNA straight in, just injecting it to make a response. And in fact, I even set up a project in my group in the early 90s, very early on, to try to immunize patients against. So what we did is we found fast ways of isolating the antibodies, and we by cloning the genes, and we were able to identify very fast the idiotype of certain kinds of antibody-producing tumors. And we thought, well, actually, let's try to generate antibodies against those antibodies, so sort of anti-EID antibodies. So we started harvesting DNA from patients, and so we knew exactly what the antibody was then they had, and then we put that DNA into patients to see if they would make a response. And really, they did make a response. This was largely a couple of medical postdocs working with me, but it wasn't good enough to do anything. That was very, very weak. Now, I think people have persisted and they've managed to improve those responses. But, it's not taken over the world, DNA vaccines. Possibly these RNA vaccines may, with, for example, Moderna with COVID, and of course, Oxford is a viral vaccine. But these vaccine approaches, which are essentially using naked RNA, I mean, this is 30 years after people were first talking about these nucleic acid encoded vaccines. So although we can often see what the next technology will be, the time scale is very, very difficult to predict. And so, for example, if you think about cellular therapy, I mean, it is not at all straightforward to be injecting cells into people. And certainly, once you have to personalize each medicine, which is to make the cells from an individual and infect them or transfect them, it becomes quite a long, involved procedure. And although you can imagine doing this in the laboratory, or actually in just sort of a series of, perhaps in a hospital, it's not something you can roll out on a huge scale. So I think that cell therapy will indeed become important. What I'm struggling with is exactly how fast it will come in. I think if it, I think there may be other ways around it. So rather than using cells, for example, instead of CAR T cells, to use adapters between T cells and target cells. So there will be alternatives which don't require producing therapeutic grade cells. So, I think cells will be important. And certainly as we get more control and more familiar with using infectious viruses that can perhaps just do one round of infection and go into cells, I think within patients, in other words, you're not having to administer cells to a patient. You're actually doing this in the body. I think that will go quite a long way. But again, I failed to predict how fast technologies would come of age. I mean, and a couple of people spent 2 valuable years of their life trying to sort that one out and failed, and we moved on. I guess if we'd stuck with it for 30 years, we might have been able to make inroads into it. But at some point, with technical approaches, you just have to make a decision that it's not, well, maybe it will be got to work, but it's probably something one has to do. And am I the best person to do it? It's probably maybe it's a small thing, or maybe it's a big thing one has to do. And so where you have a technology that relies on something erratic, you just don't know what it is. You have no particularly competitive advantage over other people in being able to achieve it, because someone else sort of thinking completely outside the box in some unknown place with no experience and not aware of the difficulties is just as likely to do it as you are. So what I tried to do was to try to focus on those things where we had got unique capabilities. And sometimes you reach a point where you try things and it doesn't really, it doesn't work, and you try a few variations and it doesn't work. So at some point you have to stop fiddling with it and you just say, well, okay, we move on. And what I don't know in the case of cell therapies is just where we are and how, yes, everything can be technically solved in my view, but how long it will take, how expensive it's going to be, what the regulatory hurdles are going to be. is another thing to think about. All the regulation, if you want to make a therapeutic, you know, you've got to try to validate an entire class. So when people talk about antibodies being expensive, well, you know, part of that is the very beginning was all of these things was a new modality, and you had to try to, you know, all the processes, including the regulatory processes, had to be developed and validated. So I think you get a new class, you'll probably have regulatory uncertainty, whatever that class is. If the FDA haven't seen it before, or the European regulators haven't seen it before, then they're going to be asking you to do all sorts of controls that actually you wouldn't get asked in with an established technology, where we think we know what signals we need to look out for, the sort of toxicity signals. There's a series of very established procedures. So don't underestimate that regulatory hurdle for bringing a new therapeutic modality in.
Great, thanks, Greg. Well, we have lots of very interesting questions coming in, but in the interest of time, I'll ask you one final question.
Okay.
Which is, if you could go back in time, what piece of advice would you give to your younger self to grow professionally, intellectually, and to inspire others to drive change?
Probably the piece of advice that I might give to myself would be I mean, of course, it might mean that one would be less successful, but I think the view would be that, I mean, if I think back to my life, the things that perhaps disappoint me about myself are that, you know, I had a number of people passing through the lab. You get friendly with them, you work with them. I probably didn't make, because I was so busy, I didn't make the effort to really get to know them all. And as I get older and I look back, I realized that some of those people were very interesting people and very nice people. And I wish I'd got more out of the people side of things as I was developing my career. But of course, if I'd done that, I probably wouldn't have been as focused as I am on the next step in what's inevitably during one's life a competitive process with which you have to survive, and you have to survive the whims of referees as well as of course all the people who could publish before you. So I would say that's probably the advice I would give myself, that I sort of probably regret not paying more attention to the people as people. But you know, I wouldn't say I was unpleasant, but I suppose I kept a distance. And but there are many other scientists who did exactly the same thing in our laboratory, in the MRC laboratory of molecular biology. And so I suppose I was following the kind of norm. Whether that would have helped me to advance the science is questionable, but it would certainly, if I look back, that's probably one of the regrets I have, that I didn't get to know some people rather better, because as I have got to know them a little bit better as the years go by, actually they're all pretty interesting. There's many things that I didn't know about them that I would have probably got something from.
Fantastic. Well, thank you so much, Greg. I know that you said you might be joining our networking platform later today. So our leaders of tomorrow will have the opportunity to ask you some of the questions that we didn't get to ask you in this session. But thank you so much. It was great to hear from you and have you here live today.
Thank you very much.